Okay, I'm Dan Janis. I'm from Charlotte. I'm going to talk about geographic and evolutionary visualization. Our goal is to develop means to analyze and share diverse types of data on organisms. All my use cases are for pathogens, but there's no reason you can't use this for biodiversity studies, metagenomic studies. It's really up to your own creativity. I'm going to use this Venn diagram to illustrate what we're doing. Primarily here, it is set in blue genomic data, orthologous sequences, okay? One to many. Evolutionary data, we use phylogenetic trees, okay? We use multiple sequence alignment and phylogenetic tree search to make sense of these data. And we use geographic information. Many pathogens now, I've had great introductions in this series, come with rich metadata, which we wish often it was richer, but oftentimes there's a place and a time of isolation. And then there's a bag of other kinds of metadata. If pathogens are from animals, were those wild animals or husbanded animals, and so forth, okay? Supermap is a web-based application, and essentially what we make is a layer for Google Earth, a phylogenetic layer. The tree is projected into the Earth, much like if I took that mobile out in the hall there and I put a big Earth under it, you would have a supermap, okay? And that layer can be shared by many users, much like we've seen before. Maybe people want to incorporate climatic data or geophysical information or transit layers to see how pathogens may be using hosts that travel along roads or flyways or so forth, okay? So it's really a framework for hypothesis generation and testing. Let's break up the Venn diagram a little bit, okay? So we have genomic data, geographic references, these sets in blue and yellow. That's really all you need. These are dirt simple file formats, FASTA files for genomic data. Don't even have to be aligned. The underlying code we use does the alignment and tree search for you. Geographic references, a CSV type format. Time is optional, okay? The movie's apparently not gonna work. I'll show it to you anytime you want. But you can also animate, interact with these visualizations as well. And many of our users are in public health service. They don't wanna work at the command line like many of Bruno's users with the command line interface. We do put out the code such that you can run on the command line interface. You can do different kinds of parameter space studies that we don't necessarily implement through the web app. But primarily people are using the web app and they get this visualization. The users are completely abstracted from PBS and all kinds of other things associated with running on a cluster, okay? The basic logic of epidemiology is the occurrence of disease, the occurrence of symptoms. Bruno also alluded to this. So epidemiologists, especially pre-genomics, may have seen the world through the occurrence of influenza-like illness, fever, headache. Your county epidemiology department right now puts out a report every month about spikes in fever and so forth. And they attribute this to flu. But many diseases cause flu, okay? You could have common colds caused by rhinoviruses in Chicago. SARS could be back in Toronto, and it's important to know the distinction between those. And in Washington, let's say, for example, the symptoms, influenza-like illnesses, could be caused, in fact, by influenza. Until we sequence the genomes of the pathogens in these three outbreaks, in this case, they are similar, let's say, for example, we can interconnect them and understand their connections to background data, okay? So here, I've did some sequencing on the pathogens occurring in patients in Chicago and Toronto, found out they shared mutations. I store that information in Common Ancestor D, but in Supermap, we also give it an evolutionary, not only an evolutionary midpoint, but a geographic midpoint. Here's some more over Michigan, for example. But we see some similarity between Chicago and Toronto and the cases in Washington. We store that information in Common Ancestor E. So we're building up a phylogenetic hierarchy projected into the Earth. What's really important here is, where does this outgroup, where does this red line connect? Can we find similar sequences in GenBank? And if so, have there been a lot of clinical studies on them? Do we know if they're drug resistant? Do we know which hosts carry them? Do we know if they're, what clinical manifestations we may have? And this outbreak can be, we can leverage knowledge to treat this outbreak right away. Okay? We can also find out from where this outbreak may be coming. We can ask ourselves, does E connect to sequences from Asia or from Europe? And I swear I made this before H1N1. Are we here in North America spawning a new disease that we should warn our public health partners in Asia and Europe or South America or wherever about, okay? So we decided, we made this framework, okay? We made this workflow, but we wanted to use it in a hypothesis-trusting framework. We wanted to use it in the context of science. So we looked into the literature, and it seemed like there was a really heated debate on which hosts carry pathogens. In the case of H5N1 influenza, many people thought that wild birds were responsible for carrying H5N1. And those people were generally those with stakes in agriculture. Must not be, poultry, it must be the wild birds that carry it. Those people who had stakes in bird conservation said, no, it must be the poultry, right? So we decided to look at this using our tool. So here's a real phylogeny, about 300 genomes of avian influenza, H5N1. And here I'm using color as well. The colors represent the host birds. Primarily blue, galliform birds, chickens, and red, ducks and geese and kin, and in white, human cases. If you drill down in the actual Google Earth file, KML file, you can see these interactions of humans and chickens in Hong Kong in '97, which was sort of the first warning that we had a zoonotic event. But clearly, you can ask the question, which birds carry avian influenza? You can address it in a context-specific way. Here you can see the common ancestor of all flu, avian flu in Indonesia, was carried over by chickens. Chickens don't fly. That clearly was trade, domestic trade in birds carrying that. Similarly, in Korea and Japan, it's carried largely by chickens, but we're honest about the ambiguity. The algorithm can't resolve the state of this character here. As we add more data, it probably can. So we're managing and being honest about uncertainty too. And here in central China, early in the epidemic, you can see a lot of ducks and geese involved. And you can drill down to any area you're interested in. This is an interesting case. Here's the same visualization. I've just tilted the Earth a little. You see this large arcing green line from Bangkok to Brussels. This was an avian flu isolate out of an eagle smuggled in hand luggage. And we think these anomalously long lines betray instances of another process, not just trade or wild bird migration, but illegal trade, or maybe even bioterrorism. What's really interesting about this virus is it's almost identical. There's no mutational difference, okay? It's genetically similar, but geographically distant. And these discordances we think are going to be very important. Here, I'm looking at avian influenza as well. There's about 600 genomes, but I've changed the coloring. I'm looking at a specific mutation here, glutamic acid at position 627 in the PB2 locus versus lysine at position 627 in the PB2 locus, the polymerase gene of avian influenza. And here the user's drilled down. He happens to be looking at a case out of a peacock in Denmark, and you can see all the mutations in a regionally specific way. But here I've colored the map because this is a key mutation that affects avian to mammalian transmission. If you've seen the reports that were actually embargoed for a long time coming out of Holland and Wisconsin about which mutations created in the lab allowed airborne transmission among avian influenza among ferrets. Okay, these are lab-created strains. What we wanted to do, we did it before, this was a low-hanging fruit, was look at how these are distributed, how these mutations are distributed around the Earth. And we can see that you can kind of get the sense of a weather map here. You have a front of mammalian-adapted viruses moving westward, okay? And we check this out with statistical validation too, okay? We don't just make visualizations and wing it. And so we started to work on the idea that the pathogens present distinct threats in various regions as a question. And here's the same kind of thing, about 677 genomes of avian influenza. And here, the color represents drug susceptibility or drug resistance. Position 31 in the M locus of the viral genome. When it's an N, it's drug resistant. When it's an S, serine, it's drug susceptible, the wild type. You can see early on in the epidemic, highly drug susceptible. Came over to Indonesia, drug susceptible. Independent of anything going on in continental Asia, within Indonesia, the virus gained drug resistance to adamantanes. And independent many different times throughout continental Asia, gained drug resistance. And when evolutionary biologists tend to see lightning striking many times at once, something's going on. There's positive Darwinian selection, and we can measure that as well, looking at what's called DN-DS ratios, ratios of non-synonymous to synonymous changes. So we have other kinds of statistical validation that coincide with the visualization. I'll show you a movie if you want to later. Then H1N1 happened, okay? This is 09, early 09. A novel, highly reassortant strain of influenza occurred in Southern California and Mexico. This is early April 09. These viruses were sequenced very rapidly and shared very rapidly with the whole world community, the data. And you can see here in about mid-April of 09, the virus is strengthening connections throughout the central US and throughout Mexico. And then about late April 09, it's getting into these megacities here in the Northeast, and that allows it to spread internationally, irrespective of US and Mexico, to Central America, okay? And furthermore, these ties are strengthening, especially between the Northeast and the Central US. This is a bit of a time series, and we thought we were doing okay. This is something we did with the US CDC, just looking at sort of Western Hemisphere viruses. But then this happened, okay? There was a global spread of pandemic H1N1 by August 2009. And although our tool behaved very well, sort of with diseases that sort of hop, skipped, and jump around Asia over a decade, these cosmopolitan diseases made a situation in which our tool didn't visually scale. I can see all the speakers before me saying, this is what we're warning about with visualizations don't make any sense. And I immediately knew this. I couldn't show this to any policymaker and say anything but, boy, it's bad out there, right? So we decided to rethink how we're showing the information. We took inspiration from airline route maps, not the ones in the back of the book in the in-flight magazine that you can't make sense of, but the ones you interact with online when you're buying a ticket, okay? So we made something called route map, same underlying data, genome sequences from influenza and lats and longs from the places from which it was isolated. But here, I was working in Ohio at the time. We allow the user to interact with places of interest. We don't show information that they don't want. And so here the user's touching on Ohio, and you get the sources of the virus for Ohio. Early on, April, May 09, you have one-way transmission indicated by the green line from Mexico to Ohio, and bidirectional transmission between Ohio, the Northwest, Southwest, and Northeast, okay? Here another user's decided that they care about the Northeast, and you can see incoming virus from Mexico and sharing with neighboring states in the Midwest, and also international exchange to Costa Rica. Here, a user decided that Michigan is of interest, and this is a little different. Michigan's a little bit isolated, has the Great Lakes around it, can't really drive across it. Data only coming in from Mexico, all right, early on. Even more isolated, no international transmission early on, is Idaho, only receiving virus from Colorado, okay? So this was really nice, even though we are trying to do things different, trying to turn over the coin of epidemiology to make it more pathogen-centric rather than symptom-centric. A lot of the modelers in epidemiology already noticed this, just working at the blackboard, right? That places that are more connected, the New Yorks, the Tokyos, are really epicenters for the spread of disease, right? And places that are isolated, the Idahos, are not, okay? So in November 09, a US policymaker asked me, what do you know about flu in the Ukraine? And I didn't know anything about flu in the Ukraine. So I went to GenBank, and there were no full genomes. There were a couple of genes. And I said, well, I'll see what's there. I wrote a couple of emails. Turns out there were isolates of flu in the Ukraine being sequenced, but it's going to take some time. This was 09. It wasn't going to happen overnight. And so we said, well, you know what? We can use this route map tool to look at the sources of flu from the Ukraine and build some medical intelligence, okay? So we used our route map tool, and we looked at Ukraine, and we found out that flu was coming into the Ukraine from New York, Ontario, Wisconsin, Catalonia, Italy, and Hangzhou in China. And we had a lot of information about these viruses, especially the ones out of New York, because New York State has essentially its own CDC in the Wadsworth Center. And we knew that We could check whether it's drug resistant. It wasn't at this time. We could also say it wasn't especially pathogenic. It was H1N1. It's bad if you're pregnant or obese, or certain young people their immune system seemed to overreact to it, but it wasn't deadly. Okay, H1N1 was successful because it wasn't that bad. Okay, it was successful in moving around the earth. I didn't know it at the time, but the policymaker didn't really care about the molecular biology of influenza. The policymaker cared because there was an election going on in Ukraine, and the opposition government and the standing government were using public health as a bit of a political football. And he wanted to know if there was really a bad flu in the Ukraine or that was political theater. And it turned out there was H1N1 in the Ukraine, but it was nothing special, and it in fact was political theater. So what we've done is now, I worked in a very computer science-oriented context for the last few years, and my colleague said, you have this really nice tool, but it's really only works with itself. Okay, what you want to do is break it up into modules and build service-oriented architectures so you can play with other users who may have different use cases or may want to bring in your service to their web browsers, okay, their projects. So what we have now is a data service. We bring in data from GenBank or your private data, georeferences. We have a visualization service. Say you don't like how we build trees, you just want to use your tree and get a KML, we'll do that for you. And primarily what we're doing, what I've really told you about is this processing service. We have a supercomputer that's completely abstracted from the user. Supermap, now among other web applications, consumes this service. You can do many different things with just alignment and tree search, right? And you can envision this as a set of Legos. We've built one toy out of the Legos, but we would hope that people take our code and build other toys out of the Legos. And I'd like to acknowledge the organizers of this meeting. This is one of the best meetings I've been at in a very long time. I see a lot of points of interface. I'd like to acknowledge the Ohio Supercomputer Center and UNC Charlotte, the American Museum of Natural History, who's been involved in this and has made the movie that I would have shown you and I will show you offline. And we have funding from DARPA, DITRA, and NSF currently. Thanks very much.